Okay, this part I believe it's like a pretty straightforward part. Uh, However, I'm going to go through it. The first group of cells is the simple epithelial or the simple epithelia. These, as we said, these are very flat cells, very thin cells, like these ones. Okay, you are looking here at a side view of them, lateral view, whereas these are superior view. So if you are looking at them from above, this is how they are going to appear. If you are looking at them from the side, this is how they are going to appear. This section is from the lungs. Okay, and here their main function is to facilitate gas exchange. Okay, that's why they are super thin, so they can allow oxygen and CO2 to diffuse in and out. Okay, so we need them here, for example, to facilitate gas diffuse or gas exchange. We can find them also in some segments of the kidney where we need exchange of materials. So we will find simple epithelia in areas where exchange of materials is needed, because we don't want much thickness. Imagine if oxygen has to diffuse through all of this thickness of the cell or multiple layers. It will be extremely hard to breathe. But because of the simple epithelia, it's much easier process. So this is the simple epithelia. Simple cuboidal, these are usually, as we said, like these are like cubes, and usually these are associated with absorption and secretions. So usually these are associated with the glands, like sweat glands, or also associated with kidneys, where there is secretion and absorption. Like the kidney example would fit with both secretion and absorption. If you want to go with the sweat gland, it's like an example of secretion. And as you can see, this is how they would appear. Sometimes in these sections, you will find like two layers of cells. That would be stratified epithelium, a stratified cuboidal epithelium. Okay? The third one is the simple columnar. As we said, these are columnar cells. Okay? And here we can find them in areas where there is a need for absorption. Like here in this example, this is in the small intestine. Why? Because we need absorption. Okay, and if you remember when we talked about the polarity of the cells, like you can notice here, all of these dark spots, which are turning yellow now, these are the nuclei. And you can see all of them are located toward the bottom of the cell to allow food, this is the food, to come into the cell. We don't need the nuclei to be here. No advantage of it being here. Okay, so this is the simple columnar. Again, in very few cases, you might find another cell or so beneath them, and that would be stratified columnar. Now, the fourth example is the stratified squamous epithelium. With the stratified squamous epithelium, as the name suggests, stratified, so we have multiple layers, okay, of flat cells. Flat cells can be seen here toward the top. Where do we find this? This is now, since there are multiple layers of cells, so this can provide greater protection. Like for example, on the skin. So technically the outer layer of our skin is stratified squamous epithelium. So now you can go and tell your friends, I can look at your stratified squamous epithelium. Our tongue also is stratified squamous epithelium. Okay, so this is where we need the protection from physical and chemical wear and tear in the skin and in the tongue. The stratified cuboidal and columnar, as we said, they are very rare in our body, so we are going to skip them. Now, another example of epithelial cells is something called pseudo-stratified columnar epithelium. Now, if you are looking at these cells, you will think these are stratified cells, maybe columnar cells. But in reality, they are not stratified. They are just one layer of cells. Just one layer of cells. And their appearance is due that their nucleus is located at different levels. So this is, for example, this is one cell here. This is one cell. Okay, this is one cell. This is another cell. You see, what's happening is that the nuclei is located at several levels, and that's why it appears as stratified. But in reality, it's not stratified. It's just a symbol. That's why they called it pseudo-stratified. Pseudo is like false. So this is falsely stratified columnar epithelium. Where do we find these cells? We can find them in the respiratory system, and mainly in the trachea. And these cells, they have cilia, if you remember the cilia from chapter 3. The movement of the cilia would cause the movement of the mucus here. Okay? So these are the cilia, and these are the pseudo-stratified columnar. Another example of epithelial cells is something called transitional epithelium. And transitional epithelium is a very unique type of epithelial cells. Why? Because they can change in their shape. Transitional epithelia can change its shape, and especially the apical layer of cells. Why do they need to change their shape? To accommodate the change of size. To accommodate change of size of the organ. For example, we have the urinary bladder. Urinary bladder. In general, urinary system, we have these cells in the urinary system, only in the urinary system, in the ureters and the urinary bladder. So sometimes we hold urine for extended period of time. If our urinary bladder is super small and can't hold so much urine, and we are accumulating urine, it will tear up. However, with the transitional epithelia, they can change their shape, okay, and that will help the organ to expand, and therefore keep urine inside, and at the same time protect the cell, protect the organ. So these cells would switch between cuboidal and squamous. Between the cuboidal, when the cell is empty, toward the squamous, to more flat cells when the urinary bladder is filled. So this is transitional epithelium. So the other part, we said epithelial cells are for lining, right? And the other thing is for glands. So all of what we covered so far was for lining. For glands, epithelial cells can form glands, either like multicellular glands or just one cell. So a gland can be like a structure made of one cell or more cells, and these cells are modified to synthesize and secrete chemical substances. And the glands can be defined or classified in two ways, as endocrine gland or exocrine gland. Let's start with the exocrine gland. It's much easier. Exocrine gland, they are exocrine because they release their content to a surface. They are exocrine because they are releasing their content to a surface, whether it's internal or external surface. Internal, like the lumen of small intestine. External, to the skin. Okay, so whether the cell or whenever the cell is secreting its secretion to a surface, it's an exocrine gland. And how does it usually do this? Because it has a duct. So group of cells, as you can see here, group of cells, they're secreting into a duct, and this duct will take these secretions to this surface. Okay, so that's an exocrine gland. Again, group of cells, they are secreting into a duct, and this duct will take the secretion to a surface. Whether it's an internal or external surface, it doesn't matter. So these are the exocrine gland. For the endocrine, simply they don't have duct. No duct. Rather, these cells are secreting into the medium, into the water, or into the extracellular fluid between them or around them. Then these secretions will be picked up by diffusion or by active transport to the blood vessels. So there is no duct to take their secretion. Okay? Usually most of the endocrine secretion are hormones. Okay? Whereas most of the exocrine secretions are not hormones, like mucus, saliva, breast milk. All of these secretions are exocrine glands. And all the glands we are going to study in AB1 are exocrine glands. Okay? So I will stop here. Again, take a break. Recap with everything regarding the epithelial tissue. Then you can come back and watch the rest of these videos about the connective tissue.